Hello everyone, and welcome to Learn While Sleeping. If this is your first time entering this podcast, just know that this is not something where you have to try it all. The goal of this is to relax, calm yourself, and be able to fall asleep naturally. There's no test, no exam, and no requirement for you to remember any of the information that we go through tonight. Everything is still going to be here tomorrow. If you enjoy this format, remember that we are also available on Spotify and Apple Podcasts. The links are down in the description. Hope you enjoy. Genetics is often called the blueprint of life, but in reality, it's less like a static blueprint and more like a living library, full of instructions, annotations, and sometimes even mistakes that can change how the story unfolds. When we study genetics, we're really asking, how is information stored in living systems? And how is it passed on? To begin this journey, let's think about scale. Imagine zooming into the human body, first to an organ, then to tissues, then to cells. Inside almost every one of those cells, in the tiny nucleus, lies an incredible amount of information. Around 2 meters of DNA, packed so tightly that it fits into something only a few micrometers across. That's like taking 20,000 books and cramming them into a shoebox, yet still being able to find any sentence that you need instantly. The language of genetics starts small. At the very base are nucleotides, molecules that are like the letters of a 4-letter alphabet, A, T, C, and G. Put them together and you form genes, which are like words and sentences. Genes don't work in isolation though. They're strung together on chromosomes, long threads of DNA wound around proteins for support and protection. A full set of chromosomes makes up a genome, the complete library of an organism. But genetics is not only about storing information, it's also about duplication and transmission. The DNA in each of your cells has to be faithfully copied and passed down whenever cells divide, and at the same time, slight variations in this copying are what create the diversity of life. Genes shape traits, but traits are rarely dictated by a single gene alone. Instead, multiple genes interact with each other and with the environment to produce the characteristics we see: eye color, height, or even risk for certain diseases. In this episode, we'll start from the very beginning: the structure of DNA and chromosomes, how we measure and organize them, and how they are replicated. Then, we'll look at the complexity of genetics, why it's not just a matter of having a gene for something, but rather a web of interactions between genes and the world around them. Let's talk nucleotides, genes, chromosomes, and genomes. To make sense of genetics, it helps to organize the building blocks from smallest to largest. At the very bottom are nucleotides. Each nucleotide is made of 3 parts: a sugar, a phosphate group, and a nitrogen base, either adenine, thymine, cytosine, or guanine. These bases are often abbreviated A, T, C, and G. Think of them as the alphabet of life, the individual letters we string together. When nucleotides are connected in long chains, they form DNA molecules. Sequences of these bases can carry instructions. Some of those sequences code for proteins, and these functional units are what we call genes. A gene is like a sentence made of letters. It has meaning, it can be translated, and it usually has a beginning and an end. Genes don't float around on their own though. They're packaged into much larger structures called chromosomes. Each chromosome is essentially one very long molecule of DNA wrapped around proteins called histones. This packaging helps fit an enormous amount of genetic information into the tiny nucleus of a cell. Humans, for example, have 46 chromosomes, arranged in 23 pairs. If we zoom out one more step, we reach the genome. The genome is the complete set of an organism's genetic material. All the chromosomes, all the genes, all the non-coding regions as well. For humans, that means about 3 billion base pairs of DNA, carrying instructions for roughly 20,000 protein-coding genes, plus many regulatory regions that help control when and where those genes are active. Let's recap that hierarchy. Nucleotides are the letters. Genes are the sentences. Chromosomes are the chapters. And the genome? Well, that's the entire book. The complete story of an organism. Understanding this hierarchy is important because many misunderstandings about genetics comes from confusing these levels. Saying that someone has a gene for a trait can be misleading. Genes work within the broader context of chromosomes and genomes, and they're influenced by the environment as well, which leads us into the idea of epigenetics, which we'll get into later. One of the most mind-blowing facts in biology is that every cell in your body, well, almost every cell, contains about 2 meters of DNA. 2 meters! And somehow all of that fits into a nucleus that's only a few micrometers wide. To put that into perspective, imagine trying to stuff 20 kilometers of string into a ping pong ball. That's the kind of compression life has to manage. The trick lies in how DNA is packaged. DNA isn't just left loose in the nucleus. That would be chaos. Instead, it's carefully wound around proteins called histones, as we discussed earlier. Those are the little bead-like structures known as nucleosomes that they form. Under a microscope, these nucleosomes look like beads on a string. If you were to take the string of nucleosomes and coil it even tighter, you'd get chromatin, a more compact fiber. And that chromatin can then be looped and folded into the highly condensed chromosomes that we usually picture in textbooks, the classic X-shaped figures that you see during cell division. There's a fascinating bit of history here. When early cytologists first stained cells in the 1800s, they noticed certain dense structures in the nucleus that took up dye very well. They named this material chromatin, from the Greek word chroma, meaning color. They didn't know what it was at the time, just that it seemed important. Only later did scientists realize that they were actually looking at DNA and proteins packed together, the very substance of heredity. This packaging system isn't just about storage, it also controls gene expression, whether a gene is turned on or off. If a region of DNA is packed very tightly, in what we call heterochromatin, the genes there are usually silent, inaccessible to the cellular machinery that reads DNA. Looser, more open chromatin, called euchromatin, is where most active genes live. So, the way DNA is folded isn't random. It's more like a library system where some books are out on the shelf ready to be read, while others are locked away in storage. And the scale of organization keeps going. If you stretched out the DNA from all the cells in your body and lined it up end to end, it would reach the sun and back hundreds of times. Yet here we are, with that information carefully tucked into the nuclei of our cells, silently guiding everything from how we metabolize sugar to the color of our eyes. The elegance of this packaging is a reminder that genetics isn't just about chemical sequences. It's also about architecture, how information is folded, stored, and accessed. Now, chromosome anatomy. This is about the landmarks of our genetic maps. When we imagine chromosomes, most people picture that familiar X shape from biology textbooks. But chromosomes don't always look like that. In fact, they only take on the X-shaped form during this very specific window, right before a cell divides. At that stage, the DNA has been duplicated and condensed, giving us two identical copies joined together. Let's break this shape down. At the center of a chromosome is a region called the centromere. You can think of it as the belt buckle holding the two halves of the X together. Depending whether that centromere is located at what position, chromosomes can look quite different. If it's right in the middle, the chromosome is metacentric. The two arms are equal length. If it's off-center, you get a submetacentric or acrocentric chromosome, with one arm much shorter than the other. In some species, the centromere is all the way at the end. These are telocentric chromosomes. Let's repeat that again. If the centromere is somewhere in the middle, the chromosome is metacentric, with equal arms of length. If it's off-center, you get a submetacentric or acrocentric chromosome, one arm much shorter than the other. Finally, some species have a centromere all the way at the end, and those are telocentric chromosomes. These shapes are so distinct that cytogeneticists, who are scientists who study chromosomes under a microscope, use them to create detailed maps called karyotypes. A karyotype is basically a photographic lineup of all an organism's chromosomes, arranged in pairs. It's how doctors can detect conditions like Down syndrome, which is caused by having 3 copies of chromosome number 21, instead of the usual 2. Now about that X shape. Each half of the X is called a sister chromatin. Sisters are identical copies, formed during DNA replication, and they remain joined at the centromere until the cell is ready to divide. When they finally split apart, each chromatid becomes a full-fledged chromosome in its own right. But not all chromosomes in a cell are identical. Humans, for example, are diploid, which means we have two sets of chromosomes, one set from mom and one set from dad. Each pair of chromosomes is called homologous chromosomes, or simply homologues. Homologues carry the same genes in the same order, but they may carry different versions of those genes, called alleles. For example, one homologue might have a version of a gene for brown eyes, while the other carries a version for blue. Here's a way to visualize the difference. Sister chromatids, they're like identical twins, formed from the same DNA during replication. Homologous chromosomes, they're like siblings. They're related, carry the same categories of information, but may have different details. The distinction is key to understanding how genetics works. During normal cell division, sister chromatids separate to ensure every daughter cell gets the same genetic information. But during the special process of meiosis, the one that produces sperm and eggs, homologous chromosomes separate, and that's what creates genetic diversity in offspring. Now, when we talk chromosomes, it's easy to assume that all living things package their DNA in the same way. But life has two very different strategies, depending on whether you're a prokaryote, like bacteria, or a eukaryote, which includes plants, animals, fungi, and protists. Prokaryotic chromosomes are simple but efficient. Most bacteria have a single circular chromosome. Imagine a rubber band of DNA floating in the cell's cytoplasm. Since prokaryotes don't have a nucleus, this circle of DNA is relatively small compared to human chromosomes, often only a few million base pairs long, coding for a few thousand genes. Despite being compact, this single chromosome contains everything the bacterium needs to survive, grow, and reproduce. Eukaryotes, on the other hand, go for complexity. Instead of one circle of DNA, eukaryotic cells carry multiple linear chromosomes, tucked safely into a nucleus. Humans, for example, have 23 pairs. Plants often go further. Some ferns have over 1,000 chromosomes. These linear chromosomes allow for a much greater genome size and more sophisticated regulation of genes. Another difference lies in packaging. Prokaryotic DNA is what you would call naked compared to eukaryotic DNA. It's associated with a few proteins, but it doesn't wrap around the histones the way our DNA does. Eukaryotic chromosomes are built with nucleosomes, chromatin fibers, and higher order folding, which allows enormous amounts of DNA to be stored in a relatively tiny space. there's also a fascinating evolutionary story here. The difference between circular and linear chromosomes is one of the clues supporting the endosymbiotic theory. Mitochondria and chloroplasts, the energy-producing organelles in our cells, both plant and animal, each have their own circular DNA, remarkably similar to bacterial chromosomes. That's strong evidence that these organelles began as free-living bacteria that were once engulfed by ancestral eukaryotic cells billions of years ago. So when you think about chromosomes, remember, bacteria keep it simple, with one circular chromosome floating free, while eukaryotes use multiple linear chromosomes stored in a nucleus. And inside our own cells, mitochondria still carry a reminder of their bacterial past. Another concept in genetics, specifically in regards to humans, is the concept of ploidy, the number of sets of chromosomes in a cell. This is one of those ideas that seems simple at first, but once you start comparing across species, it gets really interesting. Humans are what we call diploid. That means we have 2 sets of chromosomes, one set from our mother and one set from our father. Altogether, that gives us 46 chromosomes, arranged in 23 pairs. 46 divided by 2 equals 23. Diploidy is the standard for most animals because it allows for both stability and variation. Every time sperm and egg unite, you get a fresh combination of chromosomes, while still keeping the number consistent across generations. But diploidy isn't the only option. Some cells in our bodies are haploid, carrying only one set of chromosomes. These are our gametes, the sperm and egg. With just 23 chromosomes each, they're streamlined for reproduction. When they fuse during fertilization, the diploid number is restored in the zygote. Beyond diploid and haploid, many organisms make use of polyploidy, meaning more than 2 sets of chromosomes. Plants are the champions here. Wheat, for example, is hexaploid. It has 6 sets of chromosomes. Bananas are triploid, which is why they don't produce seeds. Strawberries can be octoploid, with 8 sets. Polyploidy gives plants incredible genetic flexibility and can lead to larger fruits, hardier crops, and rapid adaptation. Interestingly, polyploidy has also been a driver of evolution. Some scientists believe that ancient duplication events, where an entire genome was copied, gave rise to the complexity of vertebrates. Having extra gene copies meant that some genes could mutate and take on new functions, while others kept their original roles. It's like getting a backup copy of a recipe. One version stays the same, while the other is free to be experimented with. There's also a human connection. While we are normally diploid, rare errors during cell division can create cells with abnormal ploidy. For example, Down syndrome occurs when there's an extra copy of chromosome 21, leading to trisomy 21. These chromosomal abnormalities highlight how finely tuned the balance of ploidy usually is in our species. So, ploidy is essential. It's essentially the chromosome head count. Haploid cells have 1 set, diploid cells have 2, and polyploidy can go as high as nature dares. This is a reminder that evolution doesn't just tinker with single genes, sometimes it reshuffles the entire deck of chromosomes. Now, if chromosomes are the chapter of life's instruction manual, then a karyotype is like taking all of those chapters, laying them out on a table, and arranging them in order. A karyotype is essentially a portrait of an organism's entire set of chromosomes, paired and organized by size and shape. The process of making a karyotype began in the mid-20th century, when scientists developed techniques to stain chromosomes during cell division. They noticed that chromosomes weren't just uniform rods. They carried unique banding patterns like barcodes. By arranging them from largest to smallest and matching pairs, researchers could build a complete map of the genome as it appears under the microscope. Humans, for instance, have 22 pairs of autosomes, the numbered chromosomes, plus 1 pair of sex chromosomes, which is XX in females and XY in males. Laid out in a full karyotype, it becomes immediately obvious if something is unusual. For example, individuals with Down syndrome have an extra chromosome 21 visible as a third copy in that slot. Other conditions like Turner syndrome, where only one X chromosome is present, or Klinefelter syndrome, where there is an XXY, also become clear through karyotyping. But karyotypes aren't just for humans. They're a universal tool in genetics. By comparing karyotypes across species, scientists can trace evolutionary relationships. For example, chimpanzees have 48 chromosomes, while humans have 46. Careful study revealed that 2 ancestral ape chromosomes fused to form our chromosome number 2. That discovery provided striking evidence of our shared ancestry. Karyotypes also reveal something about chromosome anatomy. Some chromosomes are metacentric, with the centromere in the middle. Others are submetacentric or acrocentric, with off-center and centromeres giving one arm much shorter than the other. These shapes are so distinctive that cytogenetics can often identify a chromosome just by its outline. And there's a personal side too. Expectant parents sometimes see their baby's karyotype during prenatal testing. For many, it's the first glimpse of their child's genetic story. Not a photo of a face, but of the very blueprints that will guide development. In short, karyotyping is a way of making the invisible visible, turning a tangled mess of DNA into an ordered chart that tells us about our health, ancestry, and the shared history of life on Earth. Every time a cell divides, it has to do something astonishing. Make a perfect copy of its DNA. Remember, in humans that means duplicating about 3 billion base pairs accurately, and often in just a matter of hours. If you had to copy a 20-volume encyclopedia by hand, with almost no mistakes, you'd be overwhelmed. Yet cells pull this off every day, billions of times without us even noticing. The process is called DNA replication, and it's based on one elegant principle, base pairing. Adenine always pairs with thymine, and cytosine always pairs with guanine. Because of this, each strand of DNA can serve as a template for creating a new partner strand. This mechanism is called semi-conservative replication. The term means that after replication, each new DNA molecule contains one old strand from the parent and one newly synthesized strand, half conserved, half new. This idea wasn't always obvious. In the 1950s, scientists debated how DNA copied itself. Some thought the entire molecule might act as a template to produce brand new double helix, while the old one disappeared, the so-called conservative model. Others suggested a more blended method, where new DNA would be mixed randomly with old. The question was settled by the famous Mendelssohn-Stahl experiment in 1958. Using bacteria and nitrogen isotopes, they proved conclusively that DNA replication is semi-conservative, which means that each daughter molecule carries one parental strand and one new strand. Replication starts at specific points on the DNA called origins of replication. Specialized enzymes like helicase unwind the double helix, creating a replication fork, where the strands separate. DNA polymerase then comes in to build the new strands, reading the old ones as templates. The process isn't perfect. Mistakes happen about once in every 10 million bases, but cells have proofreading enzymes that correct most errors. Without those repair systems, mutations would accumulate too quickly for life to survive. It's worth pausing on how astonishing this is. Every time you grow, every time you heal a wound, your body is carrying out trillions of these replications, flawlessly preserving the instruction manual that makes you who you are. And yet, replication also leaves room for change. Rare mistakes that slip through become mutations. Some are harmful, but others are neutral, or even beneficial. In fact, evolution depends on this tiny error rate. Without it, life would be static and unable to adapt. So DNA replication is both the guardian of stability and a basic source of change. This is a perfect balance between preserving the blueprint of life and allowing it to evolve. Now, how did scientists track DNA replication? When scientists first began studying DNA replication, one of the biggest challenges was this: DNA is invisible to the naked eye. You can't simply watch strands being copied under a light microscope. So, researchers had to get creative. They turned to radioactive and heavy isotopes, special versions of atoms that act like tiny molecular tags. One approach used 32 phosphorus, or radioactive phosphorus. Because phosphorus is a key component of DNA's backbone, scientists could feed cells phosphorus labeled with this isotope. When the cells built new DNA, the radioactive phosphorus was incorporated right into the strands. By following the radiation, researchers could literally track which DNA was new and which was old. Another classic method used 15-nitrogen, a heavy form of nitrogen. This was at the heart of the Messelson-Stahl experiment, often called the most beautiful experiment in biology. Here's how it worked. They grew bacteria in medium containing heavy nitrogen, so that all the DNA became dense with the 15-nitrogen. Then they switched the bacteria to medium with lighter nitrogen, 14N, and let them replicate. By spinning the DNA in an ultracentrifuge, they could see distinct bands showing whether the DNA molecules were heavy, light, or a hybrid of both. The pattern confirmed the semi-conservative model. Each new DNA molecule contained one heavy strand and one light strand. These labeling experiments didn't just answer abstract questions. They gave us a window into the molecular world, proof that DNA replication follows predictable and testable rules. They also highlighted how powerful indirect evidence can be in biology. We can't watch atoms moving in real time, but by tagging them with isotopes, we can reconstruct the story after the fact. Interestingly, these techniques also laid the groundwork for modern medical imaging and genetic research. For example, radioactive labeling is still used in certain cancer scans, and isotope tracing helps biologists study how nutrients move through the cells. In a way, those early DNA experiments shaped both basic science and applied medicine. So, when you hear that DNA replication is semi-conservative, remember, it wasn't just an idea. It was demonstrated by clever experiments using the invisible fingerprints of isotopes, proof that even the tiniest molecules can be traced with the right tools. Now let's talk about one of the biggest myths about genetics, about phenotype complexity, and the myth of a single gene for. In popular culture, it's common to hear phrases like the gene for intelligence, or the gene for athletic ability. It sounds simple and almost deterministic. If you have the gene, you get the trait. If you don't, you won't. We often also hear phrases like the gene for blue eyes, the gene for cancer, or even the gene for musical talent. It makes genetics sound like a vending machine. Insert one gene, get one trait. But in reality, this picture is far too simple. The truth is that most traits, especially the ones we care about, like height, eye color, or disease risk, are actually polygenic. That means they're influenced by many genes working together, often dozens or even hundreds. Each gene contributes a small effect, and the overall phenotype is the result of all those effects added up. Take eye color as an example. For a long time, it was taught in classrooms as a simple Mendelian trait: brown eyes dominant and blue eyes recessive. But modern genetics shows it's much more complicated. Multiple genes control the production, distribution, and types of pigments in the iris. That's why we see such a wide range: hazel, green, gray, blue, and brown, rather than just two categories. Even when a single gene plays a large role, it rarely works in isolation. Consider the MC1R gene, which influences red hair. Variants in MC1R increase the chance of red hair. But whether someone actually has red hair can also depend on other genes and even environmental factors. This is why two people with the same MC1R variant might not look identical. And then there is the environment. Genes provide the blueprint, but environment provides the conditions for how that blueprint is read. Two individuals with the same genetic predisposition for height, for example, may end up different heights if one grows up malnourished and the other has abundant food. The same applies to complex diseases. Heart disease risk comes from both genetic variants and lifestyle factors, like diet, exercise, and smoking. So when we hear about a gene for something, it's usually shorthand. It means that a particular gene is involved in influencing a trait, but not that it single-handedly determines it. The reality is almost always a gene network, where multiple genetic and environmental inputs interact. This complexity can be frustrating if we want simple answers, but it also explains the beautiful diversity of life. No two people are exactly the same, because no two combinations of genes and environments are ever exactly the same. Well then, what shapes a phenotype more than just DNA? If genes are the blueprint, then phenotype, the observable traits that we see, is the building that results. But just like real buildings, the final structure depends on more than just the blueprint alone. Weather, materials, and construction choices all leave their mark. For living organisms, there are three big influences, which are genes, environment, and lifestyle. Genes will set the baseline. They provide the instructions for making proteins and running cellular processes. But those instructions can be interpreted indifferently depending on the context. For instance, a gene that increases the risk of heart disease might only have that effect if combined with a high-fat diet or lack of exercise. Environment also plays a massive role. Identical twins are a great example. They share the same DNA, but if one grows up in a polluted city while the other grows up in a clean rural environment, their lungs and their risks for disease may look very different. Even subtle differences in sunlight exposure, pathogens, or stress can shape how genes are expressed. Lifestyle ties into environment, but is more about choices. Diet, physical activity, sleep, smoking, and alcohol consumption all interact with our genes. Some genetic risks can be reduced, or even neutralized, by healthy habits, while others may be amplified by poor ones. And then there's chance. Sometimes random events during development, like small errors in cell division or spontaneous mutations, will influence traits in ways that can't be predicted from other genes or environment alone. One way to picture this is with disease risk. Take cardiovascular disease. Genes might set a baseline predisposition. Environment, like access to healthy food and healthcare, adds another layer. Lifestyle, such as diet and exercise, pushes the risk up or down. Together, these factors create the overall outcome. This complexity is why scientists caution against genetic determinism. Having a particular variant doesn't mean a certain fate. Instead, genes interact with everything around them to shape the phenotype we actually see. So when we talk about traits, whether it's height, eye color, or disease, we're really talking about the outcome of a conversation between DNA and the world. Let's use an example to define why the concept of saying a gene for something misleads. Science fiction loves to play with the idea of genes for extraordinary traits. In Andy Weir's novel Project Hail Mary, for example, there's mention of a supposed gene for coma resistance. The story suggests that a few people who are very lucky have this gene, and it allows them to survive long-term induced comas that would kill anyone else. It's a neat narrative device, but in real genetics, things aren't so straightforward. Why? Because very few traits boil down to a single gene. Coma resistance, if it existed at all, would almost certainly depend on dozens of interacting factors: how the body regulates metabolism, how neurons respond to low activity, how organs handle prolonged inactivity, how blood clots form, even how the immune system reacts. To bundle all of that into a single gene is oversimplifying to the point of distortion. This is where the phrase "a gene for" becomes dangerous. It suggests a direct one-to-one relationship, when most traits are multi-layered and polygenic. Even rare conditions caused by mutations in a single gene, like cystic fibrosis or sickle cell anemia, still show variation in severity depending on the environment, lifestyle, and other modifying genes. So when the public hears about the gene for obesity or the gene for alcoholism, it can reinforce a misleading idea of genetic destiny. It makes traits sound inevitable, when in reality, they're the outcome of many influences. Scientists often mean this gene contributes to the risk of, rather than this gene guarantees. Now, that doesn't mean fiction is useless. Stories like Project Hail Mary can get people curious about genetics and spark conversation. But the real lesson is this: most traits, especially complex ones, are networks, not switches. There isn't a single light switch in your DNA that you can flick on to turn on coma resistance, or intelligence, or creativity. Instead, there's a vast network of dimmer switches, knobs, and dials, all adjusting one another. So next time you hear the gene for X, it's better to imagine it as one thread in a web. Important, yes, but never acting alone. Let's discuss complex traits and polygenic variation. When Gregor Mendel studied pea plants in the 1800s, genetics seemed beautifully simple. Flowers were either purple or white. Seeds were either round or wrinkled. Traits appeared to fall into neat categories, controlled by single genes with clear dominant and recessive patterns. But once scientists began looking at human traits, the story quickly became more complicated. Think about height. Some people are short, some tall, but most fall somewhere in between, creating a continuous spectrum. There isn't just one height gene. Instead, researchers have identified hundreds of genes that each add a millimeter here, a millimeter there. Together, they produce the full range of human heights we see in population. Eye color is another good example. For years, biology classes taught a simple model: brown dominant, blue recessive. But reality doesn't fit so neatly. Many genes affect how much melanin is deposited in the iris, how it's distributed across layers, and even how light scatters through the tissue. That's why we see green, hazel, gray, and endless shades between. This type of inheritance is called polygenic, or many genes working together. The result is quantitative or continuous variation, where traits form a gradient rather than categories. It's the difference between a light switch and a dimmer dial. Mendelian traits act like switches, on or off. Polygenic traits act like dimmers, thousands of subtle adjustments creating infinite shades. And here's the twist. Even traits we once thought of as simple often turn out to have hidden layers of complexity. Freckles, hair curl, even lactose intolerance. They're influenced by more than one genetic factor, plus environment. Understanding this matters beyond curiosity. It helps explain why predicting traits or risks for diseases is so challenging. A person may carry some variants that increase risk for diabetes, for example, but without the full polygenic picture, plus lifestyle and environment, you can't say with certainty whether they'll develop it. So genetics has shifted from Mendel's neat pea plant categories to something more like a painter's palette, countless genes mixing and blending to create the infinite variety of human life. So we've covered a lot in this episode, and hopefully the pieces are beginning to fit together. Let's take a moment to step back and see the big picture. We started with the basics: nucleotides, genes, chromosomes, and genomes. These are the layers of organization, from the smallest building blocks to the full instruction manual of life. We saw how DNA isn't just loose in the nucleus, but carefully packaged into chromosomes, with architecture that influences which genes are active and which stay silent. Then we explored chromosome anatomy, centromeres, arms, homologues, and sister chromatids, and how these features help cells organize their genetic material. We compared prokaryotic simplicity with circular chromosomes to eukaryotic complexity with linear chromosomes inside a nucleus, and even tied in the evolutionary story of mitochondria and their bacterial origins. From there, we looked at ploidy, haploid, diploid, and the fascinating world of polyploid plants, and we saw how karyotypes give us a bird's eye view of the entire genome, revealing both normal variation and chromosomal abnormalities. We also dove into DNA replication, the semi-conservative model, the elegance of base pairing, and the clever experiments with isotopes that proved how it works. Replication showed us life's delicate balance, preserving stability while still leaving room for evolution through mutation. Finally, we turned to phenotype complexity. We challenged the myth of a single gene for a trait, looking at how polygenic inheritance and environmental factors create continuous variation instead of simple categories. Traits like height, eye color, and disease risk are really the outcome of gene networks interacting with the world. Taken together, these ideas form the foundations of genetics. They explain not only how information is stored and copied, but also why traits in the real world are so varied and nuanced. In the next episode, we'll move into the processes that make genetics dynamic, like mitosis and meiosis. We'll see how cells divide, how chromosomes are distributed, and how recombination and independent assortment shuffle genetic information into new combinations. These mechanisms are the bridge between the DNA library we've discussed today and the diversity of life we see around us. So as you drift off, keep in mind that inside every cell of your body lies an entire library of information, meticulously packed, faithfully copied, and quietly interacting with the environment to create the traits that make you who you are. Thank you for listening, and good night.